Hello friends, in my previous video we discussed about cell culture contamination, specifically biological contamination in details. In this video we will discuss about how do these biological contaminants enter in your cell culture, or what are the different sources of these contaminants, and how can cell culture contamination be controlled. So let's begin. Serious contamination outbreaks can occur due to improper maintenance or operation of sterilization autoclaves and ovens. If your packing material is too much compact into an autoclave or dry heat oven giving uneven heating, there may be a chance non-sterile supplies. Using too short sterilization cycle, especially for autoclaving volume of liquids greater than 500 ml per vessel, contains solid or viscous material, maybe like agar or starches, is a common mistake for non-sterile supplies outbreaks. Another thing we should care about the proper storage of autoclaved supplies or autoclaved solutions. Storage should be at dust or insect-free area to prevent recontamination. Care must also be taken to avoid condensation on bottles of solutions stored in refrigerators or cold rooms. If we talk about media or sera or other animal derived biologicals, they are not heat sterilizable and then they require membrane filtration, or sometimes radiation is also be used to remove biological contaminants. Membrane filtration through 0.2 micron is very effective in removing most biological contaminants. Airborne particles and aerosols are the greatest sources of microbial contamination in most of the laboratories. Major source of particulates and aerosols are laboratory personnel. If you wear a street clothes or dirty lab coats, which are the significant dust magnets, and placing a dust-laden sleeves into a laminar flow hood generates a cloud of dust particles that can easily fall into and contaminate cultures during routine processing. Mycoplasma contaminates your culture very easily if you are talking and sneezing, which can generate significant amounts of aerosols during routine practicing. Incubators, especially those maintained at high humidity levels, can be a significant source of biological contamination in the laboratory. Dirty water reservoirs and shells or culture vessels soiled by spilled media allow the growth of spore generating fungi. The fans used in many incubators to circulate the air and prevent temperature stratification can then spread these spores and other particulates. Some incubators humidify incoming gases by bubbling them through the water reservoirs at the bottom of the incubator. The aerosols generated by this will quickly spread any contaminants in the water. While laminar hoods and incubators are the major sites where biological contamination occurs, transporting cultures between these two sites also provide opportunities for contamination. Crawling into cultures, unsealed culture plates and dishes, as well as flasks with loose caps to allow gas exchange, provide another common way of contaminants to enter cultures. It is very easy for the space between the top and bottom side walls of a dish or a flask and its cap to become wet by capillary action with medium or condensation. This thin film of liquid then provides a liquid bridge or highway for microorganisms to either swim or grow into the culture vessels. Even without any detectable film, fungi as well as other microorganisms can grow on the outside of culture vessels. Eventually, their hyphae grow right up the side wall of the dish or past the cap into the neck of the flask. This is more often observed in long-term cultures maintained in the same unsealed culture vessel. Small insects and other invertebrates can also make temporary visits into unsealed cultures, especially dishes and plates, leaving behind only the contaminants carried on their feet. Accidents are often overlooked as a significant source of cell culture problems. Cell culture related accidents are one of the leading causes of cross contamination by other cell cultures. For example, a technician retrieved a vial labeled X from a liquid nitrogen freezer thinking it contained the widely used diploid human cell line, but instead it contains plant cell line. Because of same labeling done by another technician, this confusion was created. Another example is in the two separate research laboratories, both attempting to develop cell lines from primary cultures, shared a walk-in incubator. One lab used the acronyms HL1, HL2, etc. to identify the primary cultures they derived from human lungs cell line. The other lab worked with cultures derived from human liver, but they too unknowingly used the identical coding systems. So it wasn't long before a culture mix up occurred between the two laboratories. Fortunately, both of the accidental cross contamination cases, although serious, were caught before they caused catastrophic problems. But the question is, how many times have similar accidents occurred and not been caught? So, in brief, biological contaminants enter cultures by contact with non sterile supplies, media, or solutions. Particulate or aerosol fallout during culture manipulation, transportation, or incubation. Contaminants swimming, crawling, or growing into culture vessels. And sometimes accidents and mistakes can happen. So the next point is, how can we cell culture contamination be controlled? Here is the solution I am describing in brief. We should use good aseptic techniques to perform the cell culture related experiments or practices. We should be careful to reduce accidents. Hygiene is important to keep the laboratory clean and avoid contaminants. Routinely monitor for contamination in cell line. Use frozen cell repository strategically and use antibiotics sparingly if needed. So this is the reference slide. So this is the end of the video. Hope you like it. Thank you for listening. For more conceptual videos, don't forget to subscribe my channel and give thumbs up, share if you like this video. Stay tuned. Take care.